All right. Welcome, everybody. Thank you all for being here so early. I hope the coffee is strong enough. So my name is Camilla Margaroli. For those of you that don't know me, I'm co-chairing this session with Alessandra Bragonzi. And we wanted to give a quick introduction and why we're having this session. So this one is not working. I'm sure that all of you have seen a version or another of this graph. And this is just to illustrate that CF is changing. Patient sample collection is changing. And we're seeing a lot of heterogeneity in our patient population. And so it's really important to come up with solid models to study inflammation and infection, and then be able to translate those in the clinic. So the goal of this session is to highlight different aspects of research that question the inflammatory processes, but also inflammation. So we'll have talks that are very different in nature, but hopefully they will have a really good overview of different aspects of CF disease. So we will look at the interaction of inflammation and these new therapies that have been developed, mechanism of infection in the lung, but also other topics that are not really addressed very often, including the role of systemic inflammation in lung function, cellular senescence and the whole concept of inflammaging, but also inter-organ interplay between especially the gut and the lung, which is kind of novel. So with that, I know you guys are not here to hear me speak, so I'm gonna leave the stage to our speaker, and I hope you enjoy the session.
So, can you hear me? Yeah. Thank you, Camilla. Thank you for being here so early in the morning. So I would like now to ask Carla Ribeiro from UNC University to come to the podium. Her presentation is titled Cystic Fibrosis Airway Inflammation Enhances the Efficacy of CFTR Modulators. So
you can have your this pointer.
It's for me to point to the slides.
Here.
But
Yes. So this has the laser pointer, and it will show on the screen over here.
okay. Okay.
And this can move the slides to the right.
Okay, can you hear me?
Yes.
Good morning. I am impressed with so many early birds because I thought only the moderators and the speakers would be here. So I would like to thank the invitation to participate in this wonderful workshop. And I also would like to say that I am a co-founder of IREX Pharma. So as this audience knows very well, CF airways disease or pulmonary disease starts due to airway dehydration as a consequence of the CF defect that eventually leads to chronic infection and inflammation. And all these changes with time will promote mucus overproduction and airway obstruction. So these changes are very bad for CF patients because they increase morbidity and mortality. So again, as you know, CFTR modulators that correct and potentiate some classes of CFTR mutations have provided a game change to CF, people with CF. And before I go ahead, I'd like to say that the work that I'm going to present today is a true collaboration from my efforts as well as the efforts of Martina Gensch. And it's basically a novel area of CF research that we have developed. But before I get there, I would like to briefly review early data that led us to research in this area. So many years ago, studying endoplasmic reticulum function and morphology in CF primary cultures of 508del or also in native tissue, we found key alterations in the endoplasmic reticulum, and these include increased ER calcium storage and increased ER mass. The ER would expand. That also was correlated with increased in protein synthesis, chaperone protein expression. So basically the protein folding capacity of the ER was upregulated, and also glycosylation events. There was a lot of oxidative stress and increase in lipid production. So all these changes were very important for the pathophysiology of CF because they would lead to an increased ER, calcium release dependent inflammation. They would lead to an increased ER synthesis of inflammatory mediators, mucin production, repair factors. They would also lead to increased ROS production and a lipid-dependent signaling. So, for example, years ago, studying markers of the ER that were, for example, IP3 receptors or markers of the ER, but also ER chaperone proteins such as calreticulin, BIP, immunoglobulin bind protein, or GRP78, or protein disulfide isomerase. We found that all these markers were upregulated in native cystic fibrosis human bronchial area epithelia, implying that the function, the protein folding capacity of the organelle was augmented under inflammatory conditions. Also in vitro, we found that if we would inflame normal area epithelia or 508del area epithelia, in either way, they would have the same phenotype. And the phenotype consisted of ER expansion and increased ER protein folding capacity that would lead to an increased calcium signaling from the ER and a hyperinflammatory response. However, when the inflammation would subside, if we leave the cells long enough away from the inflammatory stimulus, they would revert back to normal. So all these findings led us to consider that the airway epithelial hyperinflammation associated with an increased ER protein folding capacity was an adaptive response to the infectious and inflammatory CF airway milieu. To address this hypothesis, we developed the following translational model. And that model consisted of harvesting mucopurulent material from excised CF lungs. That material was subject to a high-speed centrifugation. The resulting supernatant that we coined SMM, or supernatant from mucopurulent material, was then filtered, pulled from several CF lungs, and applied to the mucosal surface of naive cultures. And in doing so, we could reproduce the inflammatory phenotype that led to all these alterations that includes ER expansion, hyperinflammation, among many alterations. So, for example, if we were to expose naive cultures to SMM, we could reproduce the high expression of ER chaperones such as BIP or calreticulin. So, based on all these previous findings, we hypothesized that inflammation promoted expansion of the ER and its protein folding capacity in primary cultures of 508del, these were all homozygous cultures, that should enhance the efficacy of CFTR correctors, because now the organelle is really allowing a much better, a more efficient function of the correctors. So, in collaboration with Martina Gens, the first study that we published is shown here. We basically address the effect of the corrector VX809 in the presence and the absence of SMM exposed cultures. And suffice to say that it's very clear here, the effect of SMM was drastic. Basically, SMM upregulated the rescuing of 508del by VX809 in such a way. And this can be seen by if we address forskolin and VX770 to allow maximal CFTR function, or if you also do CFTR inhibitor 172. These functional alterations were also coupled with biochemical alterations. So basically, in presence of inflammation, SMM, we were able to see a much higher level of BNC, or the mature CFTR. But this was not just for VX809, because when we also tried different combinations of modulators, we could find that as long as SMM was present, in every case, there was a significant enhancement of 508del CFTR function. However, SMM comes from end-stage lung disease, because we have the excised lungs, and there is a lot of inflammatory, environment in that situation. So we used a second model, and that second model was basically harvesting BALF from pediatric people with CF. And the reason for that is that they would have a milder inflammatory condition. Nevertheless, what we found is that in the case here now of the corrector VX661, that BALF, similar to SMM, was also able to upregulate the rescue of CFTR. And again, you can see here, based on a increased expression of BNC, showing the correction. Interestingly enough, all these conditions that we have used, none of them were able to decrease the inflammatory status of the cultures when we inflame them either with SMM, as shown in red, or with BALF. So basically, none of the combinations of modulators that we tried in vitro were able to have some kind of anti-inflammatory effect based on iolate secretion. Now, more recently, Martin and I started studies, and actually this is now in press, in which we also tried to see what happens now to a rare mutation, the N1303K, which is not approved for ETI or the lexacaftor, tezacaftor, ivacaftor therapy. And suffice to say that ETI alone gives a little bit of a rescuing, but not enough for approval. However, when we had the presence of SMM, that combination was significantly enhanced for the ability of CFTR function to occur. Again, this can be shown here by the CFTR maximal response, or also by the response to the CFTR inhibitor 172. And here is simply a comparison of what we get if we were to do 508-del CFTR culture. So it's not so far from it. Now, as I said, SMM comes from highly infected and inflamed CF lungs, and SMM is composed of a plethora of inflammatory factors from inflammatory cells, from epithelia, from everywhere. So we decided to address, can we use in lieu of SMM a defined inflammatory stimulus? So years ago, we have addressed the inflammatory mediator profile of SMM. And of course, it's a typical Th1 profile. And what we found is that from all the cytokines, IL-8 and IL-1 beta were the highest. So we used these two cytokines in lieu of SMM. And only interleukin-1 beta, but not interleukin-8, was able to stimulate CFTR function, as shown here again by maximal CFTR response or by the inhibitory effect of 172 inhibitor. And interleukin-8, interleukin-1 beta, again, can promote biochemical alterations in the CFTR and correct much better. Interestingly, this effect is mediated by activation of interleukin-1 receptors, because when we used in similar experiments interleukin-1 receptor antagonists, the so-called NACFC, we were able to completely abolish IL-1 beta upregulated CFTR function. These are all delta F508 cultures. Again, IL-8 had no effect. But we went back to SMM, and we did the same study in which we treated the cells with SMM plus minus NACFC. And again, NACFC was able to inhibit SMM upregulated CFTR. So they are basically, at least in part, using the same mechanism to upregulate CFTR function under inflammatory conditions. But IL-1 beta and SMM also have a lot of other effects that can be common. For example, some of those ER chaperones that I have been studying earlier. We can also demonstrate in bulk-seq analysis by gene expression that interleukin beta also upregulates them, similar to SMM. So basically, these two conditions are leading to an increased protein folding capacity. So altogether, this led us to propose the following interplay between airway inflammation, increased DR protein folding capacity, and the CFTR rescue. We start with a high level of airway epithelial inflammation that would lead to an increased protein folding capacity, as we have shown in vitro. That would lead to an increased CFTR rescue in presence of the modulators, and that obviously should lead to an enhanced mucociliary clearance. But now, as the mucociliary clearance is better, is more efficient, that should lead to a decreased airway inflammatory condition, because now the noxious stimuli, they're all being flushed away. So in turn, we are considering that there should be a second phase in which is now a negative feedback, that all these alterations that I have described will now go back to some kind of normality, and then the modulators would be less efficient. while this is very difficult to test in vivo, because how we're going to do this, but there are some data coming out that can address, at least in part, that this may be indeed the case. For example, Riemann et al. In 2021 published a paper in JCI in which they were studying, they utilized baseline sputum inflammatory markers. For example, they used interleukin beta and interleukin 8, and they were correlating the change in lung function after ivacaftor. So here basically is the change in percent predicted FEV1. And bottom line is, the more inflammation there was, the more they could see an improvement in lung function. Then our wonderful collaborator, Charles Esther from the UNC CF Center, who is now a major collaborator for us in some studies that we're doing, also in getting samples from the RSCF. He has been a collaborator of the RSCF for many years now. So Chuck got bronchoalveolar lavage, and these lavages were from preschool children that were treated with ivacaftor. So what he found is that when he correlated the percentage of PMNs or neutrophil elastase with the delta pragma percent disease, the pragma score is basically a method that can allow the understanding of lung deterioration, if you will. It's a whole bunch of algorithms, morphological changes and so on. So suffice to say that the higher the pragma score, the worse the CF lung disease is. And as you can see here, there is an inverse correlation that signifies that the more inflammation, the less worsening of lung disease. And finally, this is also now, I can say, impress Chuck and his colleagues from UNC and Duke University. They studied 2 patients that harbored the N1303K mutation, and they were treating these patients with ETI, elexacaftor, tezacaftor, ivacaftor. And what they found is that these patients had a wonderful clinical benefit from the lung pulmonary function point of view with the treatment. However, when they were also doing analysis of the sweat chloride, there was no improvement. So we are trying to consider that while in the lungs there is a lot of inflammation that could have triggered those alterations I've been talking about, in the sweat glands, there is no inflammation, and therefore these changes would have never occurred to account for the lack of improvement of chloride. And finally, what is the implication of all this? Again, this is hypothetical, but I would like you all to consider. So when we start HEMT, which stands for Highly Effective Modulator Therapy, HEMT is expected to increase the mucociliary clearance, and this should result in a decreased bacterial burden, inflammation, leading to improvements in the FEV1. However, as the FEV1 improves and inflammation subsides, HEMT efficacy would then lower, and this would decrease MCC, leading to a decreased FEV1. So basically, again, it's the cycle that we believe may exist in vivo in the airways of patients. So I'd like to conclude to say that we have shown from our in vitro studies that inflammation enhances the rescue of 508del CFTR, and now I can say N1303K as well. This effect is mediated by activation of interleukin-1 receptors and couples to any protein folding capacity that is increased. Because of our studies, we believe that preclinical screening of CFTR modulators must be performed under conditions in which the inflammation and infectious milieu of CF airways is mimicked. Also, we believe that the impact of anti-inflammatory drugs on modulator efficacy should be investigated, and these should lead to optimized protocols for modulator therapy. And finally, which is also very important, lack of correlation between change in sweat chloride and change in lung function in clinical studies may be related to differences in the inflammatory status of the tissues. So this is very important for therapying, and you should keep this in mind. And thank you very much. For the interest of time, I'm not going to go in details, but again, I thank Martina Gaines in particular for this collaborative work that has become so productive and so interesting, and Chuck Esther, Charles, for all the clinical aspects of the work we are doing. Thank you.
Thank you, Carla, for the. I think it's not working.
Oh.
Hello. It's not working. Okay, I don't know. It's not working.
Should I stay or should I go?
Maybe no. You stay. The microphone is not working, so maybe we can speak together from this. And we open the discussion. Very interesting talk. So, please.
Carla, wonderful talk. Katie Heiser from National Jewish Health. I saw the original work when you showed that inflammation increased modulators, and it's really exciting to see that you're getting down to the molecular mechanism now. Knowing more about the molecular mechanism, do you think there's a role to theratype the patients in terms of, I guess, more immune phenotype the patients? Do you think we should be doing bronchoscopies to figure out, are some people not responding because their airways have different immune environments? IL-1 beta we think about as being present in the CF airway, but that's probably not in the same concentrations as you saw in the graph across the population. But so do you think there's a role to going in and trying to understand the individual immune environment in individual patients?
Of course, the more you can do a more precise medicine-oriented approach, the better, right? Because, now, as Camila has said earlier, it's a huge, spectrum of the CF patients, how they respond, what kind of lung disease they have, how heterogeneous that has been studied a long time ago. So yes, I believe that this should be a good approach to better, understand the situation of individual patients and then treat them in a much better way. Yes.
Hi, very nice work. Dan Argytes from Dartmouth. Just anecdotally, we just had a N1303 patient who started on Trikafta who had a large improvement in FEV1, but no change in sweat chloride. So it's nice to see the mechanism for how that might be working. I had a related question to Katie's. It's interesting to see that the BAL fluid had a similar response to the SMM. Have you interrogated that in terms of what may be the factors there? Because it's likely to be much more dilute, obviously, because you're instilling all the saline and then just getting a small amount of the actual lining fluid back.
Yeah, I forgot to say that when we use the BAL, we actually concentrate the BAL fourfold. We could concentrate it more, but yeah, this would be an issue that we consider because it's very diluted. But in theory, we would expect or to a certain extent, a similar cytokine profile. So,
Thank you.
Carla, may I have a question for you?
Yes,
Have you
may.
characterized SMM for bacteria, presence of bacteria cells, and in case how you consider that?
the quick answer is not really, but when we started, the SMM model, we had bacteria, basically Pseudomonas aeruginosa always, as well as in some SMMs, Staphylococcus aureus. We have not done that, but we do have a mass spec analysis of all the components of SMM that we could, so I can try to get more information for you later.
Okay. Thank you.
All right. If there are no more questions, thank you, Carla. As a reminder, you guys can submit questions through the app as well, and we can read them out loud for you. So I would like to welcome on the stage the next speaker, Jay Coles, who is going to talk about system serology in CF.
Good morning. Thank you. Thanks to the organizers. Is this advanced, or can I also just use the keyboard?
Yes.
Yeah, okay.
Yes. Do you need a pointer?
I can use that, right?
I'm not sure no.
Does it show?
No, because you have to click over there.
Oh, you got to change it to laser.
Laser pointer. Okay.
Perfect. Thank you, Alessandra. So Alessandra's already heard this talk because I was recently in Milan visiting her group. So I have no disclosures. I want to start with acknowledgments because this was truly a team effort to do this project. So it was led by this graduate student, Shiping Liu, who's now at Amgen. And then Christine Bojanowski. These are adult CF program. And then we partnered with Marlene Saavedra's group at National Jewish Health for a validation cohort study. And this actually really originated in a study that Kong Chen and actually Jeff Curlin should be on this slide at a project that we did at Pittsburgh. And then the system serology work was done in collaboration with Bonnie Gunn. So Bonnie Gunn trained with Galit Alter, who at Harvard, who Ragon Institute, who's now at Moderna. But she kind of developed this concept of system serology, which I'll introduce here. So we know from the gene modifier studies that there's a strong signal with HLA class 2. And not only in terms of lung function, but also in terms of age at first culture of Pseudomonas. So, and just to remind you what class 2 does, class 2 presents antigen to CD4 T cells and drives CD4 T cell responses, but it's also important in also generating antibody responses. So when I was at Pittsburgh, Jeff Curlin and I got an IRB approval to add a bronchial brush to clinical bronchoscopies. And compared to non-CF controls or asthmatic samples that we got from Sally Winsel's asthma program, there was a strong B cell receptor signature in the airway of CF with Bruton's tyrosine kinase and a number of B cell related genes. And moreover, there was tons of class switch to immunoglobulin genes in the mucosa of these brushes. So we modified the IRB because we only had IRB approval to take the brush and extract RNA. We modified the IRB to also get the B cells and try to understand the antigen specificity of these cells. And this shows you that this is just an example of lung function in the gene modifier study. So you can have some that are actually quite severe and then some with quite normal. And this is all homozygous F508del patients. So we wanted a systemic way to study antibodies in subjects with CF, and this is called system serology, where not only do you look at antigen specificity, but you look at the FC portion, which actually is very important in antibody function. So there's 4 isotypes in humans, 1 through 4, and they have different activity in terms of, so IgG1 is excellent at fixing complement and engaging FC receptors, whereas IgG4 is an immunomodulatory antibody, doesn't activate complement, doesn't activate FC receptors very much, and then both IgG3 and IgG2 can also activate natural killer cells. And actually, that's regulated not only by the isotype, but how much the heavy chain is fucosylated, so post-translational modification of the antibody. And the Alter lab at Ragon Institute had developed assays to measure all these effector functions of antibodies. So we decided to take a serum approach because obviously, since we were getting B cells from patients undergoing bronchoscopy, that's a clinically biased population, right? It's it's patients requiring clinical bronchoscopy, so it's skewed. And so this is the reason why we chose serum or plasma, and we basically had pre-specified criteria. So basically, homozygous F508del, age greater than 14, at least 2 positive sputum cultures for Pseudomonas, and then lung function above 90%, FEV1 above 90%, or FEV1 less than 70%, and then we had a series of non-CF controls. And then, and then we assayed both planktonic PA1 Pseudomonas, but also clinical isolates that we got from Jen Bomberger, a non-mucoid strain PA47 and a mucoid strain PA46. So if you just look at total IgG against non-mucoid Pseudomonas, you actually find levels in healthy controls. And we attribute this to what, as Alice Prince would say, is that anybody took a shower this morning and got an aerosol of about 10 to the 8 Pseudomonas. So we all have Pseudomonas antibodies, but they're higher in CF, and the total IgG didn't differentiate. But when you start looking at subclasses, there was a greater degree of IgG1 in CF patients with poor lung function. And this is just, and then we did this again with the mucoid isolate. And in this case, we saw very few antibodies in healthy controls, but obviously saw patient anti-mucoid Pseudomonas antibodies in patients with CF. And you can see that, again, there's a higher degree higher IgG1 concentration. This is a flower plot in patients with poor lung function. And then we did FACS to see if this is surface antibody staining. And you can see again by flow cytometry, there's higher IgG1 deposition on, this is again PA46, the mucoid strain, in patients with poor lung function, suggesting that there's a surface epitope that these antibodies are directed against. So to identify what these antigens are, we just basically did an antibody pull down experiment and did LCMS, and we identified a porn called OPRI, which is actually increased in biofilm Pseudomonas in terms of expression and less so in planktonic Pseudomonas. So we made recombinant OPRI and looked at levels, and you can see against OPRI, again, the antibody titers are much higher in cystic fibrosis patients, but if you do a dilution series, this is just an endpoint dilution assay, but if you titer it out, the area under the curve is significantly higher in patients with poor lung function. And this is just a statistical analysis of that. So next we worked with Bonnie Gunn to develop these assays to look at phagocytosis, C3 deposition, which is basically a measure of complement deposition, and NK cell activation. And I'm going to mostly focus, all these data are recently published in Cell Reports Medicine, so I'm just going to really focus on the complement assay because that was the one that showed the greatest difference. And so the way the complement assay is done is you take recombinant OPRI and conjugate it to a bead, and then you incubate the bead with antibody, and then you add guinea pig complement, and you can measure how much complement deposition is on the bead. And in using this assay, there was much more complement deposition when we coated with OPRI and then used sera from patients with poor lung function. So this was a discovery cohort that we used archived serum from the CF Foundation, and then so we reached back out to them to get a validation cohort. And again, in this validation cohort, we found higher anti-OPRI IgG and IgG1, again associating with worse lung function. What's interesting is in the initial training cohort, we actually saw an increased IgG4 in patients with good lung function, but this did not replicate in this second cohort. But so we thought that maybe in some patients that make a dominant IgG4 response to Pseudomonas, that may theoretically be protective. So I'm a Koch's postulates kind of guy. I say, well, this is nice human stuff, but can we replicate this in mouse? And so, and for those that aren't familiar, but in 1966, there was the RSV vaccine that was inactivated by Form 1 inactivation, much like polio. And when children in Washington, D.C. were immunized, they made weekly neutralizing antibodies to it. And then when they got RSV, they actually had worse lung disease. So that's one reason why it took almost 50 years to get another RSV vaccine, which we have now finally. So we Form 1 inactivated Pseudomonas. He killed Pseudomonas, and then we looked at strain differences between C57 and BALB/C mice. And you can see that in both B6 and BALB/C, with the he-killed Pseudomonas, that you get a strong IgG1 response. Now in the mouse, IgG1 is more like IgG2 in humans. So the complement activating isotype in mouse is IgG2C. And you can see that in B6 mice, with heat kill, we also got a high IgG2C response. So in animals that have this high IgG2C complement activating antibody response, when you infect them with mucoid Pseudomonas, they have greater weight loss and increased lung pathology. And then we also we could use adjuvants to switch the antibody response. So if you immunize with alum, you get predominantly an IgG1 response. If you use this E. coli label toxin A1 adjuvant, that generates an IgG2C response in mouse. And so this is just the ratio of the antibody responses you get. And again, if you immunize mice with and drive this IgG2C response, there's the significantly worse lung pathology. And what Xiping went on to do is transfer these antibodies into rag mice, and she could recapitulate the phenomenon. So rag mice don't have any T cells or B cells, so the only antibody they have is what you give it. So, and those mice also had worse lung pathology, suggesting that it was the humoral response to this vaccination that was driving lung disease. So what happens with ETI? So this is, so Xiping went to National Jewish and ran these anti-OPRI IgG antibodies in Mimi Saavedra's cohort where she had blood before and after initiation of ETI. And what you can see is that there's a significant reduction in these antibodies. And then the reduction was actually highly correlated with both improvement in FEV1 and also reduced risk of hospitalization. So in conclusion, I think, there's several aberrant immune responses we just heard from Carla about SMN and airway inflammation, but there's type 17 inflammation in the lung, and we've shown in prior that the B cells that we get from at least patients undergoing clinical bronchoscopy have very little somatic hypermutation, so they're largely making germline weakly avid antibodies, but their isotypes, the isotype that seems to activate complement seems associated with worse lung function in this study. So thank you very much, and happy to answer any questions.
Are there any questions from the audience?
What is the mechanism by which ETI is reducing the antibody?
So the question is, what is the mechanism by which ETI is reducing antibodies?
That's a great question. So Mimi had measured CFU of Pseudomonas, and that doesn't change. So the antibody change seems to be regardless of Pseudomonas. So so I don't know whether that's, there's CFTR and B cells, so that's one hypothesis. But there may be some other aspects of just reducing inflammation in the airway too.
Jay, can you clarify a couple of things for me, please? So one, you showed in your early data that there was increased class switching or increased evidence for genes that are class switching, and then in your conclusion you said the opposite. So that's one. Maybe you were referring to the mucosa versus the systemic circulation in terms of B cell repertoire?
So there's increased B cells, but then when you sequence the antibody from those sorted B cells, there's less somatic hypermutation.
Okay.
Yeah.
Okay.
Yeah.
Great.
So they can class switch, but they don't. The degree of somatic hypermutation is reduced.
Okay.
And patients undergoing clinical bronchoscopy.
Thank you. And then the second one is about complement expression in the BAL, because your mouse studies were, with, I guess, systemic infection, right?
But no, we did airway infection,
Airway as well.
and we did C3 knockouts. But the trouble is the C3 knockouts are so sensitive to Pseudomonas, they die. So we couldn't. So we're looking at C5A and some other complement knockouts that aren't so lethal when you give them Pseudomonas.
But your inference is that the OPR1 antibody dependent sort of effect in the patients would be mostly waged in the airways, correct?
Yes.
Okay, so you're expecting there as well that in the BAL you're going to have like some complement.
Yeah, that's a great question. So so we used to think that C3 was mostly made in the liver, but we now know that airway epithelium can make C3 locally. Harish Kulkarni and other people at WashU have shown that. So we haven't looked at complement levels in the airway. I think it'd be very interesting. We've tried to do some. So our model here is we're thinking that these antibodies are forming immune complexes in the airway, and obviously, with Pseudomonas in a biofilm, antibodies aren't going to be very effective at eradicating that anyway. So we've been trying to stain for immune complexes, but that it's been a little bit tough. We've actually got some lung samples from Scott Randall's lab, and but it's been tough to detect that. We're still optimizing the IHC. Yes.
Hi, Jess. Wonderful talk. So I'm curious. Do you think that this is something specific for Pseudomonas aeruginosus, or in general, patients with CF will have a skewed IgG response to any type of infections or allergen?
Yeah, I think it probably depends on, I think obviously microbial persistence and whether the microbe is in biofilm state. I think in planktonic infection, the Pseudomonas, the antibodies can opsonize and engage FC receptors. But I think once you get biofilm formation, potentially, and also, that there's not only bacteria in there, there's obviously a lot of DNA, host DNA, and stuff like that. That may shield the effect of these antibodies. And so we think it's almost like kind of like a lupus pathology where you get, these antibodies deposition and whether it occurs in non-CF bronchiectasis, we're not sure. We're collaborating with an investigator in Nagasaki because he has a diffuse panbronchiolitis cohort, which is where we got azithromycin from, right? So just to see if those patients show a similar immune profile because they have chronic Pseudomonas infection.
Great. Any other questions?
Okay. Thank you.
Thank you, Jay.
So I would like now to ask to Ravi to come to the podium. I short your name because it's very, very difficult to pronounce, but you can do yourself. And may I open your presentation? Oh, I'm sorry. Okay. So his presentation is entitled Erzin is required to maintain the number and function of tissue-resident alveolar macrophages in cystic fibrosis during infection. So the podium is yours, Ravi. This is, do you want the pointer?
Can I use this?
Wait, yes. Okay, this is the point.
Timer.
Is here.
Oh,
Okay, 15 minutes. Okay, you can change it from here or there or here.
Good morning, all. Myself, Ravi. I'm a postdoc in Dr. Brucia lab in Department of Pediatrics, Yale University. First of all, I would like to thank the chairs for giving me this opportunity to discuss our ongoing lab research on Erzin is required to maintain the number and function of tissue-resident alveolar macrophages in CF during infection. Regarding this project, I have no conflicts of disclosures to disclose. So before going into my talk, I would like to give a brief introduction on macrophages and their role in CF. So in non-CF lung, the macrophages play a major role in maintaining the lung homeostasis, in which they balance the host defense and inflammation in lungs. Whereas in CF lung, these macrophages are dysfunctional. Our group and many others have shown that because of this dysfunctionality, it leads to imbalance in defective host defense and hyperinflammation in the lung. In a simplistic way, at steady state, the lungs are composed of two types of tissue-resident macrophages. The one is alveolar macrophages, the other is interstitial macrophages. The alveolar macrophages are the first-line defense in clearing pathogens by phagocytosis. However, little less is known about the functionality of the interstitial macrophages, but it is believed that they are involved in immunomodulatory function. During infection or inflammation or such insults, these alveolar macrophages are partially depleted, and to compensate, the monocytes are recruited into the lung and differentiated as monocyte-derived macrophages. And over a period of time, they achieve the phenotype of tissue-resident macrophages as monocyte-derived alveolar macrophages and monocyte-derived interstitial macrophages. So in this talk, I will be focusing on alveolar macrophages. So the similar decrease in alveolar macrophages has been observed in CF patients. So this has been reported by Dr. Brito and his lab members. And they used in the CF sputum samples, they saw the decreased alveolar macrophages using single-cell RNA sequencing. Also, they saw the increased recruitment of monocytes in the same CF patients. And our group, most interestingly, we have shown that increased recruitment of these monocytes in CF mouse models has led to irreversible lung damage. So, which is why it is very, very important to study the depletion of alveolar macrophages in early stages. So we were curious to know how does this infection leads to loss of CF alveolar macrophages? What are the mechanisms involved behind it? And to study that, we incorporated a LPS-based murine model where we nebulized the mice with Pseudomonas aeruginosa LPS with a dosage of 12.5 milligram for 15 minutes. So we do this for 3 consecutive days with a period interval of 24 hours each. After the final dosage, post 24 hours, we extract bronchoalveolar lavage fluid for further analysis. So in this study, we used 3 types of mouse model. The one is wild type, the other is CF knockout model, which bears class 1 mutation. And the third model is mutated G501D, which bears class 3 mutation. So during analysis, we saw that increased recruitment of neutrophils and increased recruitment of inflammatory cytokines such as IL-6 and IL-1 beta in both the CF model post-infection when compared to wild type. So this recapitulated the CF lung neutrophilic hyperinflammation, which has already been widely known. Further, using a flow gating strategy, which is established by Dr. Oz from our group, we could able to distinguish tissue-resident alveolar macrophages and monocyte-derived alveolar macrophages. So when we looked at the wild-type mice post-infection, we could able to see the tissue-resident alveolar macrophages and monocyte-derived alveolar macrophages. It got more interesting when we looked at the CF models where those tissue-resident alveolar macrophages were drastically down compared to wild-type mice in response to LPS. And in parallel, we saw the recruitment of monocyte-derived alveolar macrophages in both the CF models compared to wild-type. So here we developed a mouse model through which we can study the alveolar macrophage, early fate of alveolar macrophages post-infection. Not only with the LPS, but when we injected Pseudomonas aeruginosa PAW1 strain bacteria through intratracheal administration, we were able to see the similar decrease of alveolar macrophages in CF model post-infection compared to the wild type. And in parallel, again, we saw the increased recruitment of inflammatory monocytes in CF model post-infection. So we were curious to know what are the mechanisms involved in causing this absence of tissue-resident alveolar macrophages in CF post-LPS challenge. So in our lab, we have been studying a cytoskeleton protein, which plays an essential role in cell adhesion and cell survival of monocytes and macrophages. So based on our previous studies, we come up with the hypothesis that loss of this cytoskeleton protein called as ezrin, it leads to cell death of CF alveolar macrophages in response to LPS or bacterial infection. So previously, Dr. DiPietro from our lab has shown that when the wild-type bone marrow-derived macrophages are treated with LPS, they were able to spread and form ezrin-rich filopodia region. However, in CF bone marrow-derived macrophages, those ezrin-rich filopodia regions were absent. Not only the localization of ezrin, but also total levels of ezrin in CF bone marrow-derived macrophages were drastically down when compared to wild-type in response to LPS. So what is this ezrin? So ezrin is an actin-binding protein which cross-links the plasma membrane and actin filaments upon activation by phosphorylation through kinases. It helps in organization of transmembrane receptors such as CFTR, CD44, ICAM1, and integrins, which are essential for cell adhesion. And through this facilitated organization of transmembranes, it helps in facilitated signaling in cell survival, cell adhesion through FAK pathway. So to know more about the importance of ezrin, we developed a mouse model where we specifically knock out ezrin in monocytes and macrophages. Using that mouse model, we isolated the monocytes and cultured them on collagen surface. Upon treatment with LPS, wild-type monocytes were able to form the lamellopodia, whereas that lamellopodia formation was absent in lack of ezrin. So due to this structural defect, when we studied the cell adhesion, we saw that lesser number of cells were attached to the collagen surface when there was no ezrin in response to LPS. And due to this defect in adhesion, we also saw that monocytes lacking ezrin were more prone to undergo cell death in response to LPS. So further, we were curious to know what signaling pathways are involved in causing these defects in adhesion and survival. So when we looked at the focal adhesion kinase and AKT signaling pathway, we saw that the phosphorylation of FAK and phosphorylation of AKT were drastically downregulated in absence of ezrin compared to wild type in response to LPS. So this shows the importance of ezrin involved in cell adhesion and cell survival of these monocytes and macrophages. Further, when we looked at the expression of this ezrin in CF alveolar macrophages, when we treated the LPS, when we treated the wild-type primary alveolar macrophages with LPS, we could able to see the formation of ezrin-rich filopodia and the cell spreading. However, the cell spreading and the formation of ezrin-rich filopodia were absent in CF knockout primary alveolar macrophages. Not only the localization, again, we observed the similar decrease in the ezrin levels in CF mouse models post-infection. And not only the total azirin levels, but the activation or phosphorylation of azirin in those primary alveolar macrophages were downregulated in CF alveolar macrophages in response to LPS. Further, we also saw that, as we saw in the lack of azirin, we saw that even in the primary alveolar macrophages, the phosphorylation of FAK and phosphorylation of AKT was downregulated in CF primary alveolar macrophages in response to LPS. So this shows the importance of azirin involved in its involvement in cell adhesion and cell survival. So now we were very curious to see why this azirin is reduced in CF alveolar macrophages. How is it reduced? So based on our previous results, we saw that at transcriptional level, the mRNA levels of azirin in CF macrophages were not downregulated. So we speculated maybe the decrease in azirin could happen at a post-translational mechanism. So based on the literature survey, we found that in CF PBMCs, the increased calcium levels were observed. Due to this increased calcium level, it might activate a calcium-dependent cysteine protease called as calpain-1. Due to this activation of calpain-1, it might degrade the activated azirin and might impair the downstream signaling pathways involved in cell death and cell adhesion. To study that, we used calpain inhibitors, where we inhibited the calpain 1 and 2 isoforms using PD15006 and calpeptin, which only specifically inhibits calpain 1. So when we used these two inhibitors, we were able to decrease the calpain activity in wild-type bone marrow-derived macrophages in response to LPS. Using the same calpeptin, when we used it in CF knockout bone marrow-derived macrophages, we were able to restore the activity of azirin. Not only the activity of azirin, but we were able to restore the philopodia region where the azirin was enriched in CF knockout macrophages. So overall, in this study, using calpain inhibitor, we were able to restore the activation of azirin and rescue the cell spreading, which led to formation of azirin-rich philopodia in CF macrophages in response to LPS. So overall, in this study, since this is still preliminary data and it's an ongoing study, so here we were able to develop a mouse model where we can study the early fate of primary alveolar macrophages over infection. And we have shown that in CF mice models, they have fewer alveolar macrophages in response to early inflammatory insults. And lack of azirin in CF alveolar macrophages reduced signaling that are related to cell adhesion via focal adhesion kinase and survival via AKT in response to LPS. And finally, we have shown that upon inhibiting the calpain-1 activity, we were able to restore the activity of azirin in CF macrophages in response to LPS. So in future, we would like to extend our in vitro study to in vivo investigation of azirin restoration in CF alveolar macrophages by administering calpeptin inhibitor, and which directly inhibits the calpain-1 and which helps in restoring the azirin. In addition, we would like to use a modulator Trikafta, where it helps in regulating the intracellular calcium levels. Indirectly, it maintains the calpain-1 activity, through which it will help in maintaining the activation of azirin, which facilitates the downstream signaling pathways to upregulate the cell survival and cell adhesion of CF alveolar macrophages. I would like to thank my supervisor, Dr. Brucia, for giving me this opportunity to work in the field of CF and for her constant support. And I would like to thank Dr. Murray for his constant support and valuable suggestions. And I would like to thank my lab members, Hassan, Kasia, Pingsha, Pam, and also I would like to thank Katrina. She is the one who started this project. And I would like to thank our collaborators, Dr. Egan, Dr. Brito, and Dr. Cross for their valuable suggestions. And also I would like to thank our collaborators, Dr. Gonzalez, Catherine, Dr. Hope, and Dr. Gupta. And I would like to thank our funding resources, NIH and Cystic Fibrosis Foundations. And all the illustrations were designed by Dr. DiPietro. And finally, I would like to thank you all. And this is us. So yesterday we were in Sedona and Grand Canyon, and now I'm in front of you giving the talk. Thank you so much.
Thank you, Ravi. So we open for discussion.
That was a lovely talk. Thank you, Katie from National Jewish Health. So you may have this data and just didn't show it because of time, but obviously if you lavage mice and you see fewer numbers, it could be because there's different numbers in the lavage, or you might not be recovering as many cells. And I know that Ezrin is supposed to increase adhesion. If you have less Ezrin, you'd have potentially less adhesion. Have you done any flow on full lung digest to see if there's actually fewer numbers of cells, or you're just getting fewer out in the lavage?
We did the flow analysis on lavage, where the data that I shown were the flow analysis data.
That's on lavage. Have you done full lung digest?
No.
Because you might just not be getting the alveolar cells out for some reason. It might not be that there's actually fewer cells.
We did do the full digestion of lung, but we did not see the significant difference in alveolar macrophages. So the major difference that we saw in the bronchoalveolar lavage.
Thank you.
Yeah.
Hi, that was a great talk. So I have
Thank
a
you.
question, or comment and a question. So my favorite protein, Splunk, inhibits calcium influx in immune cells and in airway epithelia. Splunk levels are down in cystic fibrosis. So if you tried adding Splunk, and I know people at Yale work on Splunk, so if you tried adding Splunk to the immune cells to see if that actually can restore the phenotype?
I mean, that's a great idea, but we haven't tried it so far. This is just a preliminary studies.
Oh, very exciting.
So, but I will take that into consideration. Thank you so much.
Right. Any other questions? Yes.
Thank you for your talk. How do you integrate these defects in the immune inflammation itself? Is it a protective effect on how to increase inflammation? Or is it effective because it decreases the response to bacteria?
That's what we were trying to find out. So that's exactly what we are trying to find out in future. So far, we just established a model where we can able to study the early fate of alveolar macrophages after every dosage of infection. So that will, and also after this 3 times LPS inflammation, we saw the hyperinflammatory effects indeed. But in future, we would like to explore more on that.
Thank you for the talk. So you're losing your alveolar max, but potentially, and then these are replaced by inflammatory monocytes. Yes. But the inflammatory monocytes presumably will have the same issue with calcium overactivation, kappa inactivation, Ezrin defect, right?
Exactly. Yeah, that's a really good question. That's why we would like to target this calpin-1 system so that it can help in recovering the Ezrin in all the macrophage phenotypes, which we have been studying in our lab with monocytes and as well as macrophages. So yeah.
Great. Thank you, Ravi.
Thank you. Thank you, everyone.
Okay. So our next speaker is Valentino Bezzeri, and he's going to talk about cellular senescence and inflammation in CF.
So thank you, Camilla. I'd like to thank all the organizers for inviting me and for selecting my poster. So basically, the question is, why speak about senescence in cystic fibrosis? We know that thanks to the CFTR modulators, life expectancy is improving in cystic fibrosis. And in other chronic respiratory diseases, including COPD and idiopathic pulmonary fibrosis, accelerated aging and senescence have been already found. So different stimuli can trigger cellular senescence, including the epigenetic perturbations, oncogene signals, including PTEN, which is quite cool in cystic fibrosis, elevated reactive oxygen species, mitochondrial damage, MAP kinases overactivation, and DNA damage. And this turn in irreversible cell cycle arrest, defective autophagy and mitophagy, flattened and enlarged cell shape, so remodeling of tissues and cells, resistance to apoptosis, and accelerated aging. And most importantly, a chronic inflammation described as senescence-associated secretory phenotype. And if you look at these triggering signals or outcomes, most of these issues are found in cystic fibrosis. But always bear in mind that cellular senescence is not just cell cycle arrest. We cannot describe cellular senescence by simply describing cell cycle arrest. Because cellular senescence is a complex process that involves several pathways, and for sure, P53, P21, or P16 phospho-RB cell cycle arrest is mandatory to describe senescence, but we also have to check other biomarkers, including the senescence-activated beta-galactosidase, the BCL pro-survival proteins, reactive oxygen species, and morphological changes. Senescence may have good and negative effects. It could be beneficial, arresting the progression of cancer, so arresting the cell cycle. And it could be beneficial also inducing the wound healing in tissues. However, it could be detrimental in inducing chronic inflammation, so inducing cancer in late stages, and inducing morphological changes, including the epithelial mesenchymatization. So we can see that in transient senescence is beneficial, chronic senescence is detrimental. Another, I'd like to point out also that senescence is not aging. Senescence is associated with conditions accelerating age-associated diseases, including, for example, diabetes, but has particular pathways and particular biomarkers. So if we look at the current landscape of cellular senescence, we can see a lot of biomarkers that we could investigate. And looking at what is already known in cystic fibrosis linked to senescence, we can see that, for example, some cell surface markers, including ceramide, is upregulated. Ceramide is upregulated in cystic fibrosis, and ceramide is a senescence biomarker. Chronic inflammation, so cytokines, pro-inflammatory cytokines, chemokines, and matrix metalloproteinases are already widely described in cystic fibrosis. Also, lysosomal increased activity was described in cystic fibrosis. And recently, Colling and colleagues described the morphological changes, including vimentin increased expression in lungs from cystic fibrosis. Again, metabolic adaptations are already described. Increased reactive oxygen species have been already described by Carla Ribeiro before, but also by is already well-established mechanism, and upregulated pro-survival pathways, including the resistance to apoptosis observed in primary neutrophils obtained from cystic fibrosis patients, is a fact. More specifically, senescence has been described in 3 publications so far in cystic fibrosis. One in 1979 in primary fibroblasts from cystic fibrosis patients in terms of cell cycle doubling, and another in more recently by Fisher and colleagues describing the increased expression of P16 and DNA damage markers in CF lungs. More recently, interesting paper on circular RNAs involved in senescence in cystic fibrosis has been reported. So we dissected all these pathways trying to describe senescence in cystic fibrosis. Looking at cell cycle arrest, we observed that both in bronchial epithelial cell lines, so isogenic W12 82X compared to 16 HPE parental controls, or in non-isogenic CFBE, we observed increased expression of P21 or P16. These results were confirmed also in primary bronchial epithelial cells obtained from CF patients and healthy donors, matched for age, of course, in order to avoid bias due to the aging of cultures. And we can observe that P16 is increased in primary bronchial epithelial cells. Again, we try to incubate the CFTR inhibitor 172, so inhibiting the function of CFTR in these primary bronchial epithelial cells, and we obtained an increase of P16 expression, which is in agreement with the previous results. About the metabolic adaptation, we observed that the BCL2 pro-survival, so anti-apoptotic protein, is increased in isogenic W1282X CFTR mutants compared to controls, and this finding was confirmed in bronchial epithelial cells, in primary bronchial epithelial cells. Looking at these results, BCL2 is hugely increased in CF bronchial epithelial cells compared to healthy donors. Again, we observed an increase in MAP kinases, including JNK, in COX-2, and of course NFKB. Morphological changes were evaluated, and as we reported here, the cellular shape is quite different both in isogenic W1282X mutants and in non-isogenic mutants, where the dimensions, cellular dimension, is totally different, and V-mentin expression is increased both in isogenic and non-isogenic cell models. So, according to the report by Collin and colleagues. Lysosomal activity was assayed as senescence-activated beta-galactosidase in these isogenic and non-isogenic models, and we observed an increase both in isogenic and non-isogenic models of senescence-activated beta-galactosidase. Again, this is not new for CF experts, but cytokines and chemokines, or the granulocyte macrophage colony stimulating factor, are increased in bronchial epithelial cells, in primary bronchial epithelial cells obtained from CF patients compared to healthy donors. And these are all markers of the SASP, so the senescence-activated secretory profile. Phenotype, sorry. So what we are doing now, we are trying to understand which kind of senescence is activated in cystic fibrosis. We know that there are several types of senescence, including DNA damage-induced senescence, oncogene-induced senescence, mitochondrial dysfunction-associated senescence, and oxidative stress-induced senescence. Then another type, the paracrine senescence, is due to the release of the cytokines and chemokines that can generate a loop of activation and a reinforcement of senescence. Always, you cannot exclude that these pathways there can be an interplay among these pathways, and cystic fibrosis could be associated to more than one type of senescence. So our conclusions and perspectives are that the senescence biomarkers are elevated in CF airway compared to controls. The inhibition of CFTR function in normal bronchial epithelial cells resulted in elevated senescence biomarker expression, in particular P16. The senescence might promote morphological changes in bronchial epithelial cells and trigger SASP. Senescence might amplify also lung inflammation in the absence of infection. And the effect of CFTR modulators, in particular hexacaftor, tezacaftor, ivacaftor, on senescence is still under investigation in our lab. And also the mechanism downstream CFTR functional defect that can induce senescence in CF airways is still under investigation. I'd like to thank all my colleagues, but in particular Christian Boni, who is the postdoc, who is following this interesting project with me, and Manuela Aristodor, our research assistant, who is performing with Christian most of experiments. And thanks also to our partners in Toronto, Tarini and Teo. I don't know if Tarini is here, but I appreciate so much the collaboration. And thank you all for your attention.
Are there any questions from the audience? I can start with one.
Yes.
How much do you think the chronic inflammatory response is playing a role versus the actual cellular aging in the results that you're seeing? And also, do you think that different CFTR mutations are causing a spectrum of senescence?
This is a great question. This is our hypothesis. Since we found that CFTR inhibition in normal cells induced cellular senescence, or at least induced cell cycle arrest, that is not just senescence. We have to improve our investigation about this aspect. But yes, different mutations, different class of mutations may induce different senescence. And chronic inflammation may induce accelerated aging by inducing the paracrine senescence. So it is a loop. Also in the report by Shapiro in 1979 in Fibroblast, they observed that for the first 19 cell cycles, cell doubling cells, okay, they did not observe a change between CF and non-CF. After the 19th cell cycle, there was arrest. So senescence may be sequential and improving over the time.
Any other questions? Right. Thank you. Okay, our last talk is from Alessandra, so I'll let her introduce herself.
Yeah. Okay. Okay, so thank you for the opportunity to share our results. My name is Alessandra Bragonzi, and I'm leading the Infection and Cystic Fibrosis Unit. I'm also leading the Cystic Fibrosis Animal Core Facility that is co-founded with the Italian Cystic Fibrosis Research Cystic Fibrosis Foundation. So my talk today, I have nothing to disclose, sorry. My talk today is centered on gut and lung pathology. So in the timeline of a cystic fibrosis patient, the gut pathology can begin even before birth with a condition that is known as meconium ileus. It progresses with several features, including obstruction, mucus hyperviscosity, chronic inflammation, dysbiosis, and can also lead to malignancies. In the same time, cystic fibrosis patients experience infection and inflammation in the airways that can progress with chronic damage and airway remodeling. It's worth noting that in this condition, there are several features overlapping gut and lung, including mucus hyperviscosity, chronic inflammation, and dysbiosis. So in this scenario, there are several key questions in the CF pathogenesis that has not been investigated. We do not know, for example, if pathological events in the cystic fibrosis lung and gut are distinct or interconnected. It has been shown in several pathologies that the gut play really a key role in communicating with distant organs and also influencing immunology. Of course, this scenario opened the question of crosstalk between gut and lung also in cystic fibrosis. And in particular, we are interested to know whether the gut mucosa may play a role in influencing the microbiology and immunology of the lung. So when we look to animal model and we target CFTR, we can see a consistent gut pathology that is very severe, fully penetrant, and this is evident in all species. These data really suggest that CFTR has a strong impact on the gut. If we look to the lung, we cannot see the similar picture because the lung pathology is evident in pig and sheep, but is not evident in other species. When we look to human, we have another level of complexity because we all know that the cystic fibrosis is determined by CFTR mutation, but the CFTR mutation is not the only responsible for the CF phenotype, because the cystic fibrosis phenotype is strongly influenced by genetic modifiers. And this concept is very evident for lung and gut. So, of course, this picture opens several questions and also question the cystic fibrosis mice, because we have several concerns related to cystic fibrosis mice that have been generated with genetically redundant C57 Black 6 inbred mouse strain. We all agree that cystic fibrosis mice do not mimic genetically diverse human population. They exhibit poor preclinical relevance and predictive value and often fail to translate results effectively to human. So for this reason, in recent years, we have started to exploit novel mouse model population that are known as collaborative cross mice. These mice have a different genetic background and are able to mimic the allelic diversity seen in the human population. Collaborative cross mice have been generated by Collaborative Cross Consortium several years ago, and they have been generated by crossing 8 different inbred mouse strains after inbreeding. So they are characterized by high allelic diversity and broad phenotypic variability. So we started several years ago by screening 39 collaborative cross lines with the aim to select one line where we could introduce the delta F508 mutation in this genetic background. So our screening started and we follow several criteria. For example, we screen all the lines for response to Pseudomonas aeruginosa. These results have been published by our group several years ago. We also select lines with high number of founder strain in a way to have high genetic variability, then low percentage of heterozygosity, and we also select line with good breeding performance and litter sites. This is because collaborative cross line have strong, they are difficult to breed, and in combination with the delta F508, this could be really a big issue. And then we select also line with absence of spontaneous disease because we do not want to have confounder condition. So at the end, we come up with one line that is called CC037. We introduce the delta F508 mutation by backcrossing with the C57 black 6 line. So we started characterization of this line. And as you can see here in this graph, this is survival and body weight. In terms of survival, the mutation, the delta F508 in the CC37 line is very severe because we have a complete mortality for male at 4 weeks and female around 8 weeks. In terms of body weight, we have a significant decrease of body weight, both for male and female compared to the wild type. So delta F508 in collaborative cross show fully penetral lethality and failure to thrive, as it has been shown for other mouse model. Gut phenotype is not really surprising because, as described for other species and other mouse model, we have a strong gut phenotype with a severe pathology that is mainly characterized by globet cell, more abundant globet cell, mucins that are increased in the gland and in the lumen, and increased thickness of the mucosa. And as I said you before, if we look to these conditions, they are really overlapping to lung pathology in human, not in mice. So next we check lung phenotype. And we check the lung phenotype at early stage, 4 weeks. That means at the time of wearing. If we look at the lung pathology, we could not see at the histopathological level the gross lesion. So these lesions are not really seen at the histopathological level, but we could see occasionally scatter inflammatory response in the lumen with the presence of the mucus and increase of globet cells. Then we shift to FACS analysis, and if we look at the FACS analysis, we can show that at the early stage, CF mice have an increased neutrophil presence of neutrophils in the lung, increased infiltrating monocytes and B cells, and this difference is significantly different compared to the wild type. In terms of cytokines response, we detected increased KC, BIP1 alpha, interleukin 17A, in HO mice compared to the wild type. We also detect increased neutrophil elastase and myeloperoxidase in the lung of CF mice. So from this picture, we can conclude that at the very early stage, these collaborative cross mice with the delta F5 mutation have an inflammatory profile in the lung. We also check systemic by hematological analysis. And as you can see here, we have a similar profile because at early stage, we have an increase of neutrophils at the systemic level that follow the inflammation of lung. Next, we check and we compare this line with C57 black 6. These mice have been maintained in the same condition, in the same stabulary, in the same room, because we do not want to have external environmental condition influencing the pathology. So as you can see here, the early lung and systemic inflammatory response that we could detect in collaborative cross-line were not present in the C57 black 6 line. In this case, both in the lung and in the blood, wild type and HO mice have no inflammatory response at early stage. We directly compared survival curve and body weight of these two lines. As you can see here, our collaborative cross line have a more severe and dramatic phenotype compared to the C57 black 6 line. So lethality and also the failure to thrive is more severe in the collaborative cross line. As you can see here, the body weight of the collaborative cross line HO with the mutation in the delta F508 is less compared to the C57 black 6. So next, with the intention to characterize the line at the adult stage, we start laxative treatment. And you probably know that for CF mice, we have established guideline for gut correction because the long-term survival of CF mice need the gut correction or laxative treatment. So we apply this protocol and we treat the animals with Movicol, starting from wearing. Surprising, after the laxative treatment and with the rescue of gut, we lose completely the lung phenotype. So we could not observe, both at the lung level and blood level, inflammatory response that was present at the early stage without laxative treatment. So this is evident in this graph, and this is the comparison of early and late stage with inflammatory response early stage, but not confirmed at the late stage. We can say that from this picture, the laxative treatment rescue the gut obstruction, but also probably resolve lung and blood inflammation in C57 black 6 in collaborative cross mice. So at this point, we start to hypothesize a role for gut in influencing lung and blood inflammation. So we start to check gut permeability in our mouse model, and we use Fitch-Destran. We measure the Fitch-Destran in plasma after oral administration to check gut permeability. And as you can see here, we could show that at early stage, the CF mice have a very high gut permeability, indicating that they have a leaky gut. And this condition is strongly reduced after Movicol treatment, laxative treatment at adult stage. So the gut permeability condition follow the trend of inflammation when we compare to lung inflammation and hematological analysis. So this is a condition, the leaky gut, where we could have spreading of the luminal content from the gut to other organs, so strongly influencing the other organ in the lung. So the leaky gut is a condition that can be determined by dysbiosis or overgrowth of bacterial cells. So we start this kind of analysis and we check the bacterial load in different organs, starting from gut mucosa, and stools. And as you can see here, we can demonstrate that CF mice have an significant increase bacteria overgrowth in the stool and in the gut mucosa, probably influencing the gut permeability. We also check the presence of these bacteria in other organs as trachea, lung, liver, and mesenteric lymph nodes. And we can see that this bacteria overgrowth is also present in the lung. So at the end of this talk, we have some key findings. We have generated a novel mouse model in corroborative crossed line, and this model revealed novel phenotype that were not seen in small animal CF model, and also raise fundamental questions about human CF pathogenesis. We show early inflammatory inflammation in lung and blood. We show that resolving gut obstruction leads to reduced inflammation in lung and blood. These indicate a possible role for the gut-lung axis in the early inflammatory response, and highlight also that the intestinal barrier destruction is a significant event in CF pathogenesis, possibly affecting the lung inflammation. These are the people in the front line. I want to thank all the people in my lab that over the years contributed to this work. Fabrizio, Cristina, Gloria, Davide, Nicola, Barbara, and Alessia. Other groups in our institution that contribute for genetic analysis, Anna Sofia, for pathological analysis, Francesca, and groups expert on gut pathology. I want also to thank external collaborator Giacomo for the histopathological analysis, and Alessandra at UNC for histopathological analysis in the lung. So this is a slide for our support and motivation. Our support, financial support, comes from Cystic Fibrosis Foundation, the Italian Cystic Fibrosis Research Foundation, but we are strongly motivated to go ahead, and we thanks our patients and family for continuous motivation for our work. Thank you for your attention.
Very nice talk. Any questions?
Hi, good morning. Wonderful talk. I'm Clemente Brito. I wonder if you have any more data about sex-specific differences or aging differences in the phenotype of your mice beyond the 12 weeks limit that you showed here.
So sex-specific, you mean?
Yeah, so differences between males and females, or differences between your early stage and maybe a year-old mice, if there's differences in their phenotype.
No, we do not have differences in male or female. We can only see small differences in survival because females survive more than male, but no major difference.
Cool. Thank you so much.
Hi, that was very nice. Sarah Clark from University of Colorado. I had two questions. One is, after the laxative treatment, have you tried reconstituting these mice? Do you know if the dysbiosis that you're seeing is with any gut microbiome, or there's been the development of a lot of different defined bacterial communities? Could you reconstitute with one of those that's not causing so much pathology, for example? Is it what does the gut microbiome look like? Is there an outgrowth of some of the bad bacteria, for example?
Thank you for the question. No, we have not tried. This is in our program.
Okay.
But you mean with fecal transplant, for example?
Yeah.
Yeah. This is something we want to try.
Yeah, I
in
wonder
the
if there's
future.
some bad players, driving that pathology.
The problem is that then what do? You continue to treat with the laxative treatment, because
Yeah, if you
this
could
is a,
prevent.
I believe that after this data, the rescue of gut is really a big problem.
Yeah.
Because you have no possibility to see other lung pathology if you rescue the gut.
Yeah, and my other
I believe.
question really quickly is, did you also see increased permeability at the lung? Because there's this idea that when you have increased permeability in the gut, you can also see it in the lung sometimes.
We have not checked.
Yeah.
No, we have not checked so far.
Thank you.
Hi, very interesting talk. Thank you. I have a question. So there was quite a variability in the severity of the gut permeability. And so my question is, have you seen some heterogeneity in the lung, either inflammation or CFU, like bacterial CFU during colonization? And if so, did you see a direct correlation between the severity of gut permeability and those markers in the lungs?
Yeah, good question. Yes, as you say, we have high different. It seems that there are several groups of mice with different permeability, high and low and lower. You have to think that the collaborative cross mice are not fully inbred for nature. So this is expected. So this is the reason why you need to check a large number of animals, and there is a really difficult work to work with that. They are highly variable in the gut, variable also in the lung, but overall they show a difference. I do not know if the group of mice with very high permeability are those that would go dying in the next days, and the other probably survive longer. I do not have a real correlation to the lung because there are different groups, so it's very difficult to correlate.
Hi, congratulations, Alessandra. Wonderful data and presentation. I'm Antonio Recchiuti from Chieti. A question related to the previous one. Have you tried to take the mice out of treatment with laxative and see if the rescue of the lung inflammatory phenotype or response is still abolished, or
the problem is that they die.
They die
they
immediately
die, and we have no possibility to check lung pathology because the gut pathology is fully penetrant. At 8 weeks is fully penetrant, and no possibility to check. We should try to think something different from laxative, probably. But at the moment, I have no idea how to go ahead and to check the adult stage without laxative.
Yes, because
Well,
it's very
any
interesting
suggestion?
idea. Very it opens very I don't know, probably like diluting the laxative like to some doses that are not very effective. So something like that.
Yeah.
Hi, thank you, Wanda O'Neill, UNC. With the increase in microbial evidence in the liver, have you checked liver pathology? I know within the CF liver field right now, there's this thought that increased gut permeability can lead to bacterial products moving through the portal vein to the liver and interacting with CFTR there to produce liver disease. And so this model might be a good place to look for liver pathology, just a thought.
To be honest, at the gross level, we could not see liver pathology, but I welcome any expert for liver. We are not a really expert. At the gross level, I cannot say we could see liver pathology. I agree with you. It's very interesting to see that we have bacteria, coli, Escherichia coli enteric bacteria in the liver, probably because of this gut permeability and the spreading of luminant content to other organs. And I think this is really important point to consider. Yeah.
I have one last question for you. Given this gut permeability, how do you think CF patients are avoiding undergoing sepsis?
So it's not really clear whether the gut permeability, it probably there is low bacterial load transfer, but we really don't know if this is the case. But there is also spreading of molecules produced by bacteria. It's not only a question to spread bacteria, probably also inflammatory molecules that go to other organs. I have a macrobiological background, and after this data, I asked to the clinic, but do you find enteric bacteria in the lung? And they said yes, but we don't mind. I'm not sure that we probably we should mind, because probably there is something that starts before Pseudomonas aeruginosa that give a sign to start inflammation, and then it comes other things. I don't know. We should think about.
All right. Thank you all. Thank you to the speakers.